Hello and welcome to today's lecture. So, in the last class we had discussed about unfolding of fibronectin and how you can go about designing experiments to determine the unfolding signature of individual domains within fibronectin 3 module. Okay. So, if I were to write down the steps, okay, you have to engineer multiple protein constructs. Okay. So, I wrote it as A B of n, where n is a variable. So, n is chosen, is n is experimentally determined so as to ensure the engineered protein folds properly. Okay. So, by doing this and choosing multiple different constructs, you can determine the folding signature of multiple domains. One thing I introduced in the last class, briefly spoke about, is rate dependent stability. Okay. So, what do I mean by rate dependent stability? By rate dependent stability, I mean that by changing the pulling rate, the unfolding force changes. Okay. And how does it change? So, more often than not, what you will see, okay, okay. So, this is, let us say, at a given tip speed, okay. So, you have what you are changing is this tip speed. Okay. So, if this is, let us say, 5 nanometer per millisecond, this is 0.5 nanometer per millisecond and this is 0.05 nanometer per millisecond. Okay. So, this axis is frequency, this axis is force. Okay. So, if you plot it for multiple different domains, so this is tip speed or pulling rate. Okay. So, you will get, okay. So, I have drawn representative rate dependent forces for 3 different domains. In this case, A is maximally sensitive to pulling forces, pulling rate, and C is insensitive to pulling rate. Okay. So, for the case of fibronectin, for example, it has been observed that FN3, first domain of FN3 is maximally sensitive to forces, and the 10th domain, okay, let us see, D is not but FN3 10th domain. So, 10th domain of FN3 contains the RGD sequence. So, this is on an average less sensitive to pulling forces. Okay. So, what if a cell is exerting so much force that the entire protein folds? Will that be the end of function of that protein or not? Okay. What has been observed is there is another aspect, okay, and you can do that. What it says that if you release the tip or hold it in one position. Okay. Okay, if release the tip after partial pulling, what will you see? In other words, okay, so let us say this is a pulled configuration from this point. Okay. Okay, the tip goes down and then you again pull. Okay, you again pull. Okay. So, what you are introducing it is, you would see that this time allows, when you relax the tip, okay, release the protein, okay, it allows the protein to refold. Okay. And how do you know what is the signature of refolding? So, let us say, okay, okay. So, this is, okay, this is the protein signature, right, protein unfolding signature. Okay. Let us assume if delta t, which is 0.1 second. Okay. After that, if you pull again, what you will see is long no folding signature, then something like this. If you pull the protein after 1 second, your signature might be, okay, and 10 seconds later you might have. Okay. So, here you see that this is indicative of partial folding, and here the protein is completely folded. Okay. So, you can plot time delay, percentage of peaks. So, what is percentage of peaks? So, what you see is that how many peaks do you get. So, here this value beyond a certain time delay, this is going to be 100%. Okay. So, if your time delay is negligible, you would see a low value, let us say 10%. So, that would still indicate that one of the peaks has completely folded, okay, completely refolded. Okay. So, what you see overall is that the complete unfolding. So, for FN3 domain module, so complete unfolding okay, would lead to extension from 160 nanometers to 950 nanometers. Okay. That is the increase in length that you observe even for unfolding of one FN3 module. Okay. One FN3 module will lead to extension from 160 nanometers to 950 nanometers. Okay. What are the other things? So, how do the unfolding forces look? So, you have seen that unfolding forces, okay, unfolding forces have magnitude order, let us say 100 to 300 piconewtons, okay, and it is known that cells can exert forces order 200 piconewtons. So, this would imply that fibronectin does get unfolded. Under physiological conditions. Okay. The last point. So, what does unfolding achieve? So, domain unfolding So, in a sense what it is modulates and elasticity. Okay. And of course, the signaling. If I were to draw force peak number, so at the initial ones, okay, so you will have, okay, you will have a linear force curve, okay, increase in forces, and among the very first ones which unfold is certain Fn3, okay, your Fn10 which contains the RGD sequence has some intermediate force, okay, and among the highest forces domain 1 and domain 2. Okay. So, these are among the highest forces. So, what you see is a great increase the amount of unfolding force, and forces regulate the downstream signaling cascade as well as the effective elasticity of the protein. Okay. So, with that I conclude our discussion on ECM proteins and how nonlinear elasticity is ingrained either at the level of a network, ECM network, or at the level of single proteins. Okay. So, as part of reading assignment, I would like to assign the following two cases. Okay. So, please read these two papers, you will get a good understanding about fibronectin unfolding. Both these papers focus on fibronectin unfolding, and most of what I discussed just now over the last 2-3 lectures comes from the first JMB paper. Okay. So, that concludes our discussion of ECM proteins. Now, I will again come back to our analogy of cell as a tent. Okay. You have cell as a tent. Okay. So, we discussed, so there are 4 things, right, plasma membrane, okay, ECM, and cytoskeleton. Okay. So, there are these 4 things. Okay. So, plasma membrane, ECM, focal adhesions, and cytoskeleton. Okay. So, these are the 4 things which are required for stabilizing cell shape. So, of these we discussed ECM, and now we are going to start discussing focal adhesions. Okay. What do focal adhesions do? So, these focal adhesions are nothing but dynamic fixtures to the ground. Okay. So, now we know that for adherent cells, so if I draw the cell, okay, the and you have these. Okay, so, this is your nucleus, this is your focal adhesion, this is Fa, and outside you have the extracellular matrix. Okay. So, if we ask that what kind of function these focal adhesions must serve, so we know that for adherent cells the cell must remain stuck to the substrate, okay, but at the same time it should be in a position to migrate. Okay. So, the cells should be physically coupled and be able to migrate. Okay. So, what kind of requirement, okay, does it put on the design of the fixtures? Okay. what can I say about the fixture design? That is FA design. Okay. So, since the focal adhesion acts as the real link from the inside and the outside, okay. So, if you had it as a single protein, so you would need for the cell to move, you would need to disengage the cytoskeletal as well as the ECM network it is connected to at all times, but that would be energetically very unfavorable. Okay. So, connection mediated via single protein is energetically unfavorable. Okay. So, there must be some other signal, the way that focal adhesions must be designed must be such, must be such that, so the design must be such that cytoskeletal does not need to be fully dismantled as cells move. Okay. So, if I were to zoom in to the focal adhesion, okay. So, this were your focal adhesions. Okay. Okay. So, if I zoom into this structure, what I propose, okay. So, this is inside the cell and this is outside. So, the design of the focal adhesion probably might have some transmembrane protein linked to some cytoskeletal protein, and then you have the connection to the nucleus. Okay. So, you have two entities, entity A, okay, which span both the inside and the outside of the cell, and interacts with the ECM. Okay. And you have another entity B, okay, which interact with A and connects to the CSK is short for cytoskeleton. Okay. So, in this way, when the cell migrates, just decoupling of B from A can allow A to again depolymerize or come off and re-engage the outside, and then as B connects, you can have this dynamic turnover. Okay. So, these fixtures or focal adhesions serve as the eyes of the blind cell, and unlike the way I simplified its design, the focal adhesion does not comprise of one cell, okay. It has greater than 100 proteins. Okay. So, you can have more than 100 proteins localized at focal adhesions, okay, and the focal adhesions serve various functions, okay, more than 100 proteins Okay. So, you will also have a broad distribution of sizes, okay, ranging from 0.5 to 1 micron square, okay, and 3 to 10 micron square. Okay. So, clearly you see that there is two different size distribution of these additions, okay, and they are present at various locations within the cell. Okay. So, what are the functions of these focal additions? So, FA apostrophe S is short form for focal additions. Okay. So, they form the physical role, that is stabilize the cell or stabilize cell shape, and actually they are the ones which actually sense the surroundings. Okay. And how do the cells sense the surroundings through focal additions? By means of force transduction. Okay. So, you can have pulling forces, that is contractile forces, you can have pushing forces, okay, pushing forces, and you can have remodeling. So, pulling forces or contractile forces are what induce these wrinkles in cells, right. I found seen this presence of these wrinkles, and that is what pulling forces induce, wrinkles in the matrix. Where would be an example of pushing forces? Okay. So, one case of pushing forces might be in the structures of invadopodia. Okay. So, it is not necessarily a focal addition structure, but invadopodia contains many proteins at invadopodia, similar to that of focal additions. Okay. So, in contrast to focal additions which are formed on the plane, an invadopodia actually protrudes into its matrix. Okay. So, if this is the matrix, okay, so these are invasive structures where cells actually protrude into the matrix. So, you can imagine a pushing force being exerted. Okay. So, that completes our initial understanding of what focal additions are. So, I said that there are lot of proteins. Okay. So, there are lot of proteins which are present in focal additions. And so, one of the most important proteins is integrin. Okay. So, integrin is the one which is present transmembrane, okay, which is present across the membrane. Okay. So, you have an integrin. Okay. Integrin is composed of two subunits. So, these are alpha and beta, okay, and these subunits are non-covalently linked. Okay, depending on the type of ECM protein. Okay. So, if I were to look at some of the combinations of integrins, okay. So, here I have drawn, what I have drawn in the center is beta 1, and what you see is beta 1 is common to many of the integrin combinations. Okay. So, you can have for all these collagen receptors, you can have alpha 1 beta 1. So, for binding to collagen, okay, you can have alpha 1 beta 1, alpha 2 beta 1, alpha 10 beta 1 and alpha 11 beta 1. Okay. Similarly, for binding to RGD, you can have, okay. So, for RGD binding, you can have alpha 5 beta 1, alpha 8 beta 1, and also you have alpha V beta 3, alpha B beta 5, so on and so forth. Okay. Similarly, you can have various other combinations for binding to laminin. Okay. So, in total there are 18 alpha and 8 beta subunits. Okay. So, you have multiple different things, for example, in fibroblasts, in most cancer cells, alpha 2 beta 1, alpha 1 beta 1 are very common. Okay. With that, I stop here for today, and I will continue in next class discussing some more of the focal adhesion proteins and then discussing about their structural position or location within the focal adhesion. Thank you for your attention.